All right, I think we're good to kind of go ahead and get started. My name is Jamie Smith. I'm one of the co-chairs of the CCHABs group, and we're excited to have you for today's quarterly meeting, the summer one. And I have another co-chair, Sarah Ryan, on the line. Sarah, you want to introduce yourself quickly?
Hey, good morning. I'm Sarah Ryan, the Environmental Director for the Big Valley Band of Pomo Indians and one of the co-chairs. Nice to see you all.
And Sarah and I are pleased to announce that we, as many of you remember, Becky stepped down at the last meeting. Becky Stanton, from formerly of OEHA, now with Department of Public Health. She stepped down as longtime co-chair, and we held a nomination period, and we received nomination for Karen Odkins, who will be joining us as co-chair, as our third co-chair. As CC has, we typically have three. So Karen, you want to introduce yourself really quickly?
Sure. Yeah. Thanks, guys. I'm Karen Odkins, as she said. I'm the Freshwater and Estuarine Harmful Algae Bloom Coordinator for CDFW, and I look forward to working with you all.
Awesome. And, incidentally, Karen has the first agenda item for today. So we'll start with some announcements. Karen had a planned kind of announcement / introduction of what CDFW is doing for their FHAB activity. So I'll go ahead and stop sharing that screen, and
Thanks.
Karen, go ahead and take it away.
All right, let me figure out how to share my screen really fast. Been a minute since I used Zoom. A Teams lady now. Let's see. Share screen. Host disabled participants from sharing screen.
Oh dear. Let's see. How about now?
Try. Oh yeah, we're good. All right, can you all see it?
Looks great.
Okay, cool. Yeah, so thank you all. So I kind of already told you my name and everything, but today I'm going to be talking and updating you guys on CDFW's illness response. So I'm going to be kind of discussing our resources, our processes, our communication lines. And I think this is actually really particularly timely. As many of you know and are working on currently, there's a bloom happening in San Francisco Bay again. So hopefully this will kind of set all of us up for this bloom that's currently happening, and give you guys an option to ask some questions if you have any. A bit of background about me, as you may know from me running for this position and sending out my resume. I was at water boards before this in the FHAB program for the Central Valley Water Board, but I just started at CDFW at the beginning of the year, so I've been here for a couple months now. Next slide. Okay. Okay. Can you guys see the next slide? Are we good?
Yes. We got it.
Okay, great. Thank you. I think it's slow on my end, maybe. Okay. So, as many of you know, AB 834, Assembly Bill 834, started the FHAB program in 2019. We do lots of different things in that program, but today I'm specifically going to be talking about the part that's event response, really coordinating the immediate and long-term event response and communicating the risks associated with that. So from a CDFW perspective, our goals for incident response are really to determine which fish are dying because or ill because of HAB-related events. So that's kind of in line with what the illness work group's goal is. Prioritizing water bodies that can be managed, fisheries can be managed in relation to HABs, and then also managing HABs that occur on CDFW properties, which of course we have some responsibility for. So in the event of a fish and wildlife event, my role is really to track cases and mortality statistics. I serve on the illness work group. I coordinate kind of the interagency response that is happening around that. I coordinate sampling when it's relevant, and also do some sampling myself when it's relevant, and then address the public media inquiries that might arise because of that particular event. So just for a little bit of context of what we're working with here, we all at the FHAB program have very limited resources. Water Board has 5 paid staff and many borrowed staff that's kind of borrowing time from other programs, and they have a small but a useful routine budget. At CDFW, it's just me. I'm the statewide coordinator, and I don't have any programmatic funds. So that's pretty limiting, but doing the best I can with what I kind of have. But recently, I was able to secure some short-term drought funding, which really was changing what I was able to do. I also have been getting some more experience over the last couple of months dealing with these different incidents. So because of those 2 things, I've changed some of the workflow that we have at CDFW, which is why I'm sharing with you today what those changes are. So with some of these funds, I was able to purchase just some really basic programmatic supplies that we really needed. So, some bottles and materials to do some data collection, safety equipment for our teams that are going out to collect information, and some really basic water quality instrumentation. Also, really excitingly, I was able to set up some lab contracts. So these are going to be really familiar for those of you who have worked with the Water Board. So it's with Ben Genetics, and we're able to send water scum samples, benthic algae mat samples. And then really excitingly, they also have a subcontract with Green Water Labs there, and that we will be able to send tissue samples, organ samples, stomach contents, and lots of things that'll help us determine fish and wildlife association with HABs. And we'll be able to get those tested for cyanotoxins as well. Because it's short-term drought funding, unfortunately, the contract ends pretty soon, March 2025. So, we'll have to use it up before then. Hopefully, fingers crossed, I'll be able to get some more funding before the end of that to continue this and create a new contract, but there's no guarantees. So I'll just walk you through what this kind of workflow looks like now. So I'm only really involved when there's a fish or wildlife illness or death. So when we receive those reports, they'll come to me. And then I'm going to look for evidence of a HAB. Sometimes people report what they think is a HAB, and it might not be. Maybe it's pollen on a lake. You guys are all familiar with things that people think are HABs that aren't. And then we're also going to look that it meets our incident criteria for sampling, and I'll go over what that is on the next slide. If it meets all those criteria, then I'm going to go and collect water or biological samples, depending on what's relevant for that situation. If it's a water sample, it'll go off to Ben Genetics. And if it's a biological sample, it has a couple pathways that it could go. So these blue bubbles here that you see, those are things that are sort of out of my control. They're at CDFW, but they're not funded by FHAB. So if they have capacity within their workday, they may take on our cases. And so those kind of options are for an autopsy with our wildlife or fish health lab at CDFW. And then they have the option of sending some of their samples off to UC Davis to test for certain diseases. So whether or not we send it off to those labs, we can also send samples off to Green Water Labs for cyanotoxin analysis. So these really have bolstered our analytical opportunities and gives us really, great understanding compared to what we had before. It takes, in terms of the illness work group, it really takes cases from kind of, saying not HAB related to a really definitive diagnosis in some cases. So I'll just give you kind of an example of one recent case that we had where that was something that happened. So a CDFW fish biologist was out doing their thing, and they were able to report that there was goldfish and carp dying in large numbers at a lake. That lake had a known HAB history, so we did suspect that it could be HAB related. He collected a specimen while he was out in the field, and we got that approved for autopsy from the wildlife or from the fish health lab. And so that was delivered there. It was a really fresh sample, and they were able to get it there quickly, which was really great. So then they looked at it there, and they decided that some of the markings on it, they would send it off to UC Davis. And they were able to test via PCR for 3 different viruses there. And they confirmed that actually they did die of koi herpes virus. So they were positive for that virus. So we were able to say that that was probably not HAB related. It was probably related to that virus. Okay, so what are those incident sampling criteria that I mentioned earlier? So again, those are just for those kind of the green bubbles that you're seeing now. So just to kind of say when we will pay for those. So we're looking for mass die-offs or illnesses in most cases. Sometimes, there's one fish that dies or, not a huge impact. And so most of those cases we won't take. When species of interest are affected, so if there's a threatened or endangered species, we're going to, of course, be really interested in following up on that. If it is a HAB priority habitat, so if it's something, a sensitive area, then we're going to be really interested in following up with that. CDFW property, of course, like I said, we have responsibility for that property. And also sampling, I kind of wanted to add something in that if there's another case that I haven't quite thought of yet or haven't encountered yet, where it really forwards our mission as an agency, we will of course look into that as well. And of course, we have limited funding, so as long as funding lasts. I've also been working to create some new SOPs, standard operating procedures, that relate to fish and wildlife HAB testing. So as you can see, I'm still working on some of these, but I have now cyanotoxin testing procedures for tissue as well as for stomach contents. And I'm still working on the ones for urine. And then I also am trying to make a really preliminary kind of rough field autopsy SOP. Autopsies are really tricky. You really have to know kind of the species that you're working with and have reference for what they look like in a healthy sample to be able to really tell what's going on in a diseased sample. But I'm hoping that we can do just some really preliminary work in case the health lab can't take certain cases. Okay, and I'm also doing a lot of training internally. So I think I've done 11 presentations internally so far for staff, just trying to teach them about HABs, trying to get them ready for incident response. We have a couple more planned this year. I'm sure more will come up as people encounter more HABs. So those kind of trainings are really about messaging a centralized sort of reporting location for folks, making sure that they know what to do in the event of a HAB, establishing a network kind of like Waterboards has of designated trained regional staff that's ready to go when there's a HAB event, and making sure they have the equipment they need. And then also establishing a backup HAB coordinator. If I am sick or if I'm out, I don't usually take time off during the summer, but if something happens, I want someone to be there to represent CDFW and to kind of be representing the fish and wildlife. And finally, I have been doing a couple other things just to refine our response process. So I've been creating some draft media releases so that, when large incidents like we're having now come up, we'll be ready to go. We've been defining some of the jurisdictions that we have within CDFW, which was an issue that we kind of learned about last year. Like I said, I was writing some more SOPs, and I also am working on collecting water quality and biological data. And also working on some record keeping that is specific to, the fish and wildlife focus that we have at CDFW. So thanks so much. That's my update. I'll stop sharing. If you have any questions, happy to take those now.
Yeah.
Or I don't know if we have time.
Yeah, I think we're out of time for
Okay.
questions, but definitely time later.
Okay, cool.
Later
or
on.
shoot me an email.
Yeah.
Thanks.
Yeah, thank you so much, Karen. That was really great. We should have given you your own specific agenda item for that, because that was really, really informational. So yes, please feel free to reach out to Karen with questions. We can hang on the line at the end of the meeting if you guys want to speak with her as well. So moving on to the next agenda item, we have Amy Jacoby from the CDC. And so Amy, you should have screen sharing privileges if you want to start to pull your presentation up. But Amy's going to talk to us about some public health research that she's been working on to understand public perception of harmful algal bloom signs. So thank you so much for joining us today, Amy.
Thank you. Yeah, trying to find my unmute button here. So let me try to share my slides and let me know if you're seeing the right side of these.
Awesome, looks great.
Okay, great. So thank you so much for having me this morning, and apologies for my voice. I have a little bit of a cold this week. But my name is Amy Jacoby, and I am a health communication specialist at the Centers for Disease Control and Prevention, and I work in the waterborne disease prevention branch. And today I'll share some information about what the public knows about harmful algal blooms using results from a nationally representative survey of US adults. And before we start, I wanted to acknowledge that developing this survey and analyzing the results was very much a group effort. So I've also listed the names of other CDC colleagues here who were instrumental in getting these results ready to share. All right, so to give you an idea of where we're headed, first I'll talk about the goal of the survey and the methods used. Then I'll spend most of our time walking through the results, and I'll wrap up with some key takeaways, next steps, and time for discussion and any questions. But before all that, let's start with some context. We all know HABs are happening in freshwater and marine water across the country, and that there are efforts by many organizations to educate people about HABs. But there are limited data about awareness among the public and the public's behaviors around HABs. There we go. So for this survey, we wanted to get a better idea of people's perceptions and behaviors around HABs, because this type of data can help us better reach the public with relevant illness prevention information. So we used 10 survey questions that broadly fell into the themes of awareness, risk perception, behavior, information seeking, and knowledge. So we used a Porter Novelli survey, which is a nationally representative survey of US adults, and a little over 1,000 people participated in early September of 2021. And the results are weighted to reflect the US population by sex, age, region, and race, ethnicity, and formal education. So now I'll walk through the results of our 10 questions, and I'll present the questions in the order they were asked to survey participants. So first we asked which of these words or phrases people had heard before, and saw that about 40 to 50% of people had heard of red tide, toxic algae, blue-green algae, harmful algae, or an algae bloom, with red tide being the top choice. And then fewer people had heard the term harmful algal bloom, and the least number of participants had heard of terms that included cyanobacteria in the name. And we also asked which terms people thought sounded like something that could make them sick. And here we saw that about 3 out of 4 people agreed that harmful algae, toxic algae, and harmful algal bloom sounded like health risks. And people were a little less likely to agree that terms related to cyanobacteria sounded like they could make you sick. And they were least likely to think algae bloom and blue-green algae sounded harmful. And next we asked who people would expect to tell them that lakes, rivers, or oceans were unsafe to go into. And most participants thought local or state officials or beach or park staff would provide this information. And they were least likely to expect this information from social media influencers and friends or family. We then asked how likely they would be to go into a water body with signs of poor water quality. And we saw people were generally unlikely to go into water with signs of a HAB, but that about 10 to 20% of people would enter water that looked or smelled bad, with people most likely to enter cloudy water. And then next, we wanted to know how severe people thought most illnesses caused by harmful algal blooms are. And over half of participants thought most illnesses would be severe enough to require emergency care or hospitalization, with very few people thinking illnesses would be very mild. We then asked another question about risk perception and saw that participants thought that all groups of people were susceptible to illnesses caused by harmful algal blooms. Although if we break those data out by likely and somewhat likely, we do see that more people thought that adults were only somewhat likely to need to seek medical care for harmful algal bloom related illness, whereas participants thought the other groups listed, including children, older adults, or those with pre-existing conditions, were likely to need medical care. So participants had a somewhat lower risk perception for healthy adults compared to the other groups. And next, we asked people what behaviors they thought could prevent HAB-associated illnesses, and saw that people were most likely to choose looking for visual or olfactory cues, such as the water looking discolored or smelling bad. And they were less likely to choose checking for fishing, shellfishing, or swimming advisories before visiting the water. We then asked about who people would report symptoms to, and most people said they would report symptoms they thought were caused by water in a lake, pond, river, or ocean to their local or state health department, followed by park or beach staff and healthcare providers. And then next, we wanted to see how many survey participants visited natural water bodies. So we asked how often people visited during the summer season. And here we saw that about 40% of respondents typically visited at least one type of water body between July 4th and Labor Day. And then finally, we asked participants if they had ever had any experiences with harmful algal blooms, and they could choose from several direct experiences, such as canceling a trip because of a harmful algal bloom, or kind of more indirect experiences, such as hearing about a HAB affecting drinking water supplies. And here we saw that almost half of participants have not had any experience with harmful algal blooms. And among those that did have experience, the majority had experiences that were secondhand rather than direct. Okay, so that was a lot of data. So I'd like to wrap up with some of our group's key takeaways from the survey. For HAB terminology, we saw that people were most familiar with the term red tide and terms that included algae like toxic algae or blue-green algae. And they were somewhat less likely to recognize terms that included bloom and least likely to recognize terms that included cyanobacteria. And the top terms that people thought sounded like something that could make them sick included the words harmful or toxic. And then for illness prevention, most respondents agreed that using visual and olfactory cues were a good way to avoid HABs and prevent illness. And fewer, although still a majority, thought that checking for water advisories before swimming, fishing, or shellfishing could help prevent illness. And then when asked about who they thought would provide information about water bodies that were unsafe to go into, people generally expect this information to come from state and local authorities. And similarly, people said that if they became ill after going in or near a water body, they would be most likely to report their symptoms to their health department. And then for risk perception, we saw that most people thought illnesses caused by HABs were severe or very severe, and that they thought HABs could make anyone sick. Although the risk perception for healthy adults was somewhat less than for other groups like children or those with pre-existing conditions. And then finally, for behaviors around HABs, we saw that about 40% of people visit natural water bodies during the summer, so they could potentially have a recreational exposure to HAB. We also saw that people weren't all that familiar personally with harmful algal blooms. About half had no experiences with them, and very few, less than 15%, had a direct experience, like actually visiting a water body during a HAB or taking steps to avoid getting sick from a harmful algal bloom. And we also saw that most people would avoid water with signs of poor quality, but still about 10 to 20% of people are likely to go into water with signs of a harmful algal bloom or poor water quality. All right, so what are we planning to do with these data? First, our group is using these results to better understand who we might need to reach with harmful algal bloom information and what kind of messages will resonate with our key audience based on their familiarity, behaviors, and risk perceptions. And we're also working on sharing and discussing these results with partners so you all can use them as well. And then we're working on collecting and analyzing some additional data to get a more complete picture of public awareness of HABs and answer some of the questions that these survey results raise. All right, so on this last slide, I wanted to put a plug in and note that CDC has HAB communication resources available for you to use. We have a variety of outreach materials, including fact sheets, posters, graphics, sample social media messages, clinician reference cards, and web pages that we encourage you to use and share. And you can find all of these materials on our website, cdc.gov/HABs. And then also please feel free to contact our group at ohhabs@cdc.gov, or you can get in touch with me directly at ajacoby@cdc.gov with any comments or follow-up questions. And with that, I think we have some time for questions and discussion. And I certainly would be interested to hear how these national survey results compare to your experiences with public awareness of HABs here in California.
Great. Thank you so much, Amy. That's really super interesting work. Do we have any questions from the group? I see a couple people raising their hands. So Shannon, if you want to unmute, go ahead and ask your question.
Thanks, Amy. So great. I really appreciate having this kind of work done. It's such important and yet underrated and underestimated type of information, because if people don't recognize the words that we're using for our messaging, the messaging is lost. In California, certainly because HABs are very prevalent and we have a very water-oriented culture and a lot of water bodies, which not every state has those two elements. There is certainly an increase in HABs. What we've seen over the last year is a decrease in illnesses, particularly with dogs. But we've seen an increase in people being proactive about either being aware of a potential exposure or calling about signage before recreating so that they can prevent an illness. So that really helps to support and bolster our messaging efforts. One note, I just wanted to echo what some of your findings were showing, that when signage is appropriately posted and very visible, that does dissuade people from recreating in waters where they could be at an increased risk for an illness. Also too, with the signage, if people do experience an illness after recreating and they did see some sort of concern or warning or algae alert, they're more likely to report it, particularly in California with the poison control. There are several cases we investigated this year that were not necessarily symptomatic or symptoms occurred later, but they were proactive in contacting poison control, which then forwarded it to the HAB illness work group through the OEHA contract, so that we could follow up with those individuals and see if they did become symptomatic, because some of them just really had second thoughts or regrets after recreating, and we're not showing illness signs, but we're worried that some could arise. And we've seen cases where they've been asymptomatic days after, and some that they do show some dermal symptoms. And again, so signage and messaging is really critical. So thanks for this work.
Yeah, thank you so much, Shannon. Really interesting to hear all you're doing in California. I know you all are a very busy group, so thanks for sharing that.
You have other questions for Amy? Let's see, Sarah, I noticed you put something in the chat. Do you want to share real briefly about what you posted?
Sure. I put the wrong link in first, but I over on Clear Lake, which is the largest natural freshwater lake entirely in California, we did do a HABs perception survey in 2021 through a project. It's actually a CDC grant that were the on the ground folks working with Tracking California. And so you can see the survey results in the second link that I sent, and we were able to incorporate just elements of the ambient monitoring program that we have on Clear Lake, and also the fact that we have tribal consumption and tribal uses. So we really wanted to check in with with our survey, or with people who we were surveying, about the impacts of HABs on tribal traditional activities. So it seems somewhat similar in terms of, not a lot of people have been impacted health-wise as far as they know, but they've either, some of them have heard about it, some of them have been impacted. There's always a lot of, there's more outreach that we can all do, so there's less confusion, I guess. But I really appreciate this survey and presentation, Amy. I think it's really useful, and I want to get a copy of it so I can share it with our CalWatch folks.
Sure, thanks, Sarah. If you'll send me an email, I'd be happy to send these slides over to you so you have them. And thanks for sharing the work you're doing locally. I'm very interested to take a look at that.
Thanks.
Great. Any other questions for Amy? Let's see. All right. I'm sure you're going to get, a couple of emails, but after the fact, as this is, we're working, on signage and various things as a CCHABS group. But this is super helpful. Thank you for your time, Amy, and for joining us and sharing your work. So with that, I'll kind of call upon our next speaker, which is Chris McClemens. Awesome. Hi, Chris. So you should have some screen sharing capabilities. So let's just make sure that works, and then you can go ahead and take it away with your presentation.
Perfect. Yeah, can you see my screen there?
Yes, looks awesome.
All right. Move you guys out of the way here. Okay. Yeah, so thanks, Jamie. I'm Chris McClemens. I'm a PhD student here at the University of Oklahoma in the lab of Dr. Dave Hambright. And today I'm going to present a little about our work on microcystis. We've done a lot of genomic and we're beginning a lot of physiological profiles of some genospecies that we've recently identified. So yeah, there we go. So microcystis, I think most people are probably familiar, very common Hab-forming cyanobacterium. It's globally distributed in freshwater systems, but also borders on submarine and brackish systems. It's found in at least 108 countries and spans 280 degrees of longitude and 90 degrees of latitude. So it has a really big range, a huge distribution going from tropical Brazilian strains up to some cool Swedish strains that we've isolated in the past. As global climate change continues, conditions are going to shift towards those that are going to increase Hab formation and its intensity and duration that they're occurring. It's also going to move microcystis into more and more lakes, and particularly interesting to us is it's going to move into lakes that aren't typical of microcystis Habs, that are really eutrophic. We're seeing it pop up more in eutrophic systems, and we even know that microcystis is inhabiting some oligotrophic systems. So problem that's not really going to grow away, only probably going to get worse. And those Habs are incredibly disruptive to freshwater ecosystems. They're impacting carbon and nutrient flows up the trophic web, and many are toxic to both humans and wildlife. These are kind of the big reasons that we're interested in studying microcystis, right? We need to figure out what's going on with it, try and get a handle of it, but it's really difficult to study, or at least study well and communicate between different groups. Microcystis has a really broad physiology. We have some that are kind of the best example is non-toxic or toxic. They have different nutrient uptake kinetics and different preferences, and they inhabit different niches. So we know there's a lot of diversity in microcystis, so it's really interesting to study and figure out what's going on there. This is clouded by, let's see if I can get my laser going. Yep. By the fact that the taxonomy is currently based on morphology, and that morphology is highly plastic. It can change. For example, if we have microcystis in the image up here in panel A, looks like a microcystis aeruginosa, it might take that colony morphology under environmental condition A, for example. Once the environmental conditions shift into another, they might change to something that looks like we have an M here, probably microcystis viridis. It's the same microcystis, but it's just changing its form to respond to what's going on in the environment. And that can make it difficult for two people studying, multiple groups that are studying the same microcystis to communicate about what's going on, because now we've given it two different names. We can't effectively communicate about what's happening. So because of the diversity that we know exists in microcystis and its ecological importance, a former postdoc in our lab, Dr. Haiwan Kai, set out during the pandemic to start to evaluate genomic differences that might be hidden within the microcystis genus as a whole. We know that historically there have been over 50 morphospecies described, so there's a lot of names given to microcystis. But again, that morphology that we're using to name microcystis is super variable, and it depends on the environment. So it gets really cloudy and noisy about what we're actually talking about when we're naming microcystis and we're studying it. Many groups have tried to use standard genetic markers, subunits of the 16S rRNA gene, ITS, CPCBA, to try and use sequence-based approaches to classify what microcystis is in a system. These unfortunately lack much consistent agreement, so it's really difficult, again, for us to name what the microcystis is in our system and get it below the genus level. Definitely good to tell us we have microcystis, but beyond that isn't really all that helpful. Others have tried to use the full-length 16S sequence, but when comparing those, they're over 99% similar. So those would indicate we have a single species. And further, when people apply other genetic similarity approaches, just looking at comparing sequences to sequences of the full genomes, these all indicate that microcystis is a single species within the genus. So people have put out that, hey, we have one microcystis species here. Maybe we have different ecotypes, essentially meaning maybe we have different strains of microcystis in the genus that confer different ecological types, forms, preferences. They inhabit different niches. This, again, gets a bit cloudy because how do you name those or classify them just on their ecology? And so we hypothesized that because of the diversity within the microcystis genus, using whole genome analyses may enable us to resolve the microcystis taxonomy and lead us to a consistent classification. And so this was the work that our postdoc started and that I have continued on. We've looked at all the publicly available genomes in NCBI and some others that we've gotten from collaborators. The initial work that I am showing here, we had 228 total microcystis genomes that I narrowed down to 139 distinct, unique microcystis. Looking at their genome size, we saw a lot of variation from about 3.9 million bases up to 5.9. So we know there's a lot of difference in the different microcystis that we've isolated and sequenced. There's a lot of diversity there, things that might be going on and might enable us to resolve differences in the microcystis that we have. There's really two sets of genes that microcystis have that will be important as we move forward here. One is the core genes. These are the genes that are shared in all of the microcystis that we have isolated. And then another set of genes are the pan genes or flexible genes. These genes are not in all the microcystis, and they often confer some sort of advantage in an environment. So they're selected for when they're advantageous in an environment. So microcystis isolated from the same or roughly the same ecological conditions probably share similar flexible pan genes. So in the analysis that we did, we took all those genomes that we have, we found all those genes, and we built some phylogenies. Here I'm showing the core gene phylogeny. It has a paired pan gene, those flexible gene phylogenies. And what we looked for were groups in these two trees that shared the same members and formed coherent groups. As you can see by the colors and the numbers, we found 25 conserved groups that are distinct, seemingly unique entities, at least at the genomic level. This gave us a foundation, a stepping off point where we can now sort of reliably classify these different groups and identify what microcystis belong in them to then begin studying their different ecologies and move forward on trying to resolve the diversity within microcystis. Just kind of a high level survey of that work and some of the things we're looking at. We first looked at if we have geographic patterns in microcystis. What is its biogeography? Do we have all the North American microcystis grouped into one group, all the Asian, all the South America? Oftentimes, we didn't find that. There were a few cases. We have group A, B, and P. They were all from North America, the US and Canada. We had groups K, N, and 20. Those were all from East Asia. But beyond those groups, we had members from different regions all shared within the same groups. So we saw very little geographic coherence, very few biogeographic patterns. This suggests that microcystis either lacks barriers to dispersal, or I think more likely that these groups in different regions are experiencing similar ecological conditions and are therefore going through a form of convergent evolution so that they are classified the same way, but are from distinct regions. We then looked at what genes they share or don't share within and between groups, and we found some differences in cytotoxic genes, but more interestingly, in some of the oruginosin and microcystin profiles, we found that for microcystin, groups generally either had or didn't have the ability to produce microcystins. This kind of binary nature is really interesting, that these groups are doing things differently and reliably within their groups. And so about half of the groups that we've identified are toxic and half are non-toxic, but only the group G that we've identified had members that did or didn't have the ability to produce microcystin. So that was a interesting kind of binary condition. That was a leaping off point into our physiology work that I will talk about in a minute. And so at this point, we looked at, we have these genospecies, potentially species that are defined solely by their genomes. There's obviously more work that we might need to do to classify these as true species, but we were now at this point kind of poised to start looking at these groups, these genospecies, and figure out, do these conform to an ecotype idea? Like, do these different groups have different ecologies? Do they fit into different niches reliably and distinctly from one another? So I went and I looked around our tree and I found different microcystis that still are in high quality culture collections, primarily the Nies collection in Japan. And I ordered microcystis from as many different groups as I could. They're all from Japanese lakes, so we were limited on our ability to look at a biogeographic spread, but we could at least find different microcystis that have different evolutionary profiles and different ecological profiles. We then got those in, we grew them up in culture, and we began studying them under different nitrogen, phosphorus, and light conditions to see if they have different ecologies. And so this work was primarily done by our collaborator, Dr. Max Berthold at Mount Allison University in New Brunswick, where we started with our culture full of media, super eutrophic. We grew up microcystis to a really healthy density. We then put it in media, excuse me, that was absent of any of those nutrients to get them to use up their reserves so that when we begin experimenting with them, they should all be starting at the same baseline. Any nutrients they're bringing in will be distinct to their kinetics, not based on what they have stored in their cells and in their community. And so we took those starved microcystis, we put them in a gradient of 4 phosphorus levels through phosphate and 5 nitrogen levels through nitrate. Those are on a factorial gradient, so we could really look at the interaction of both N to P, phosphorus, and nitrogen alone. And we took those nutrient gradients and we then put them under 2 light levels to see how their growth rates change. Over a period of about 2 weeks, we measured fluorescence and absorbance to start getting growth rate data, and we analyzed that data in R to get growth rate kinetics and begin some niche profiles. And so just an image of their liquid handler here, but I think important to note is these are in the bottom right, the starved microcystis. And what we initially noticed, and I observed this in our cultures as well, is that these microcystis are handling the stress of being starved, but also just growing under the media in different ways. So we are already, before collecting any data, observing that microcystis are handling nutrients differently and growing differently alone. So here's an example of some of the data we collected. This is for our Nies 88 strain. And when we're looking at this plot, we have cell densities on the left, time, or on the y-axis, elapsed time on the x-axis. And as we move left in these figures, we're increasing the phosphate. And as we go down in the figure, we're increasing nitrogen, so that the bottom right panel is showing hypereutrophic conditions. As much nitrate and as much phosphate as we gave them is down on the right. Essentially no nutrients of control in the top left then. And we had our 2 light conditions colored by the 2 colors, but in this data and this form, kind of a a course level look at it, light did not really seem to have an impact. This culture and many of them, they acclimated to both light conditions well and grew equivalently between light. As we increased nutrient availability, perhaps somewhat surprisingly, it took longer for the cells to get to a stationary phase to use up their nutrients. That makes sense. And nitrogen was actually the main driver of biomass production. Phosphates seem to have little effect. As we go left to right in these panels, there isn't much difference in these curves. They're growing the same amount, they're reaching the same densities at each phosphate level. But as we go down, we're getting higher and faster growth with more nitrate. This would make sense. Microcystis doesn't have heterocyst, it can't make its own nitrogen. So giving it more makes sense that it's responding more that way. Digging into the data a little further, trying to profile these microcystis that came from different groups, we normalized then the amount of cells that we got at the end of the experiment to the amount of phosphate that we gave it. Since they didn't respond to phosphate, we normalized our cells to the amount of phosphorus we gave it. And in doing this, we began to observe some differences in the different groups of microcystis that we identified in our genomic work. The 5 strains on the left and in the middle, they all responded well to increasing nitrogen, meaning they grew more with more nitrogen we gave it and increased the rate that they grew at. The strains that produced microcystin, are 2 on the left here, ended up having the highest production of cells over the course of the growth, generally over 1 million cells per micromole of P that we gave it. So these are really strong growers. Our microcystis in the middle, microcystis 98 from our genus species G. This one was one of the only ones that seemed to respond strongly to low light, whereas the others responded either not at all to light or responded better to high light. So we had over the amount of nitrogen added, we see an uptick here in the amount of cells we get, the amount of growth we have. Whereas under the highlight, it doesn't change much. We just have a stable growth, doesn't really care how much nitrogen we give it. As long as it has some and that high light, it's just going to grow and hang out. Our other 2, 2481 and 3804, there was a weaker effect, some effect, but it was weaker with more nitrogen, and there was no light effect at all in these treatments. And so just from these 5 data points, these 5 different strains from different groups, we're beginning to resolve that these microcystis are growing differently. They're responding to their environment or different elements of the environment. And just a note, our 2 final strains all the way on the right, these either didn't respond to our treatments or ended up dying during the course of the experiment. So they weren't happy with really anything we gave them, whereas our others were fine and happy to grow. The last thing that we started to look at was optimal N to P, trying to get into some niche modeling and figuring out what makes these microcystis tick, where do they like to be in the environment. We only had enough data from 4 strains to do this modeling, but from this, we observed that our strain from Utex grew highest around 30:1 N to P under the low light, but under the highlight conditions, we actually didn't reach an optimum yet. It's somewhere above 60:1, where it would prefer to grow best. Our NES 88 was growing best around 40:1, but we observed higher growth under the highlight condition. Our NES 98, we again saw our growth optima around 40:1, but it preferred low light conditions. And our NES 2481, we had the highest growth under 40:1 N to P, and was slightly faster under highlight, but it seemed effectively indifferent to what light we gave it with regard to its N to P ratio. And so again, we're finding that these different groups, when we start to model them under their different nutrient conditions and getting towards niche modeling, these microcystis do have different growth preferences, different growth conditions, and potentially using a single species concept or just calling microcystis is probably not effective when thinking towards management decisions and how to best manage microcystis moving forward. And so what we're getting out of this work and moving towards is that we know microcystis is phylogenetically, evolutionarily distinct within all the members of this genus. We have different metabolic genes, different characteristics throughout these different groups that may then confer differences in their growth that we're starting to observe in our physiology experiments, at least with regard to nitrogen, phosphorus, and light. These might then push us towards what ecologies are feeding back into the genomic differences we're having. Why is the environment that these microcystis are in shifting their evolution, how they're grouped together, and making them different from one another? We still have a lot of work to go to continue to profile the genetic characteristics of microcystis and their physiology to begin to try and link what genes they have to their environments and the feedbacks that are going on there, especially since this is a growing problem. What role microcystis is having, even if it's negative in the system, will be helpful to know at an ecosystem service level. And finally, I think one of the kind of bigger takeaways that I'm moving towards and exploring is that given the variability in the response to nitrogen, phosphorus, and light, we're beginning to think that single nutrient management solutions, so remove just P, remove just N, may not be applicable for all microcystis. So understanding what makes each microcystis tick and what's in a system and classifying that reliably may be important moving forward so that we're properly handling our system, so that we're decreasing microcystis that we have in a system, not potentially pushing it towards favoring another. And so with that, kind of a high overview of our work, I'd like to acknowledge some of our collaborators and our funding sources over the past few years. And also a bit of a request that if you have microcystis in your systems or even cultures that you've isolated, we only have one microcystis in the database from California right now. It'd be great to have some more to see what's going on in North America. I'm seeking microcystis cultures and scums from around the world, as well as here in the US. I'm aiming to establish them as cultures and then sequence them to, again, try and fill in gaps in our tree and begin to do some niche profiling from as much diverse microcystis as I can get my hands on. So I'll leave my email up here and the email of my advisor, Dave Hambright. So reach out if you have some microcystis, but with that, I'm happy to take any questions on our work, what it means, where it's going, go from there.
Thank you so much, Chris. That's a fascinating presentation. I'm seeing a couple people in the chat that have already sent you some questions as you were talking. Okay. So Hans Pearl put in the chat, he said, did your microcystis cultures grow as colonies or as single cells or both?
Yeah, hi Hans. So in general, our cultures are growing as single cells, but we have two strains that when they get to stationary phase, they become colonies. In the physiology experiments, though, they were kept moving and they did not form colonies during the physiology experiments. They were stopped before they got to that formation during stationary phase.
And Hans, did you have any follow-up for that, or that answer your question? Feel free.
No, this is, yeah, I don't know if you can hear or see me.
Yeah.
I'm over here sitting in a dark place. The reason I asked was because of the light situation. You know, the impacts of light when the colonies, well, the colonies are going to self-shade themselves to some extent. So that may be an issue with the experiments with light.
Right.
But I think, by the way, your talk was great. And I really, it confirms a lot of the field work that we've been doing with microcystis blooms that basically it takes two to tango. You got to have both N and P in there to really soup up the bloom. And I think your experiments really confirmed that, which is really nice.
Yeah, we're excited to keep them going and see what we can profile here.
Thank you.
Yeah, thanks, Hans.
All right, and then Kelly in the chat asked, are you beginning to look at the impact of microplastic that the microplastics might have on microcystis?
Yeah, that's a really interesting question. Thanks, Kelly. So I personally am not, and our lab is not. However, that is an area that is seemingly a direction people are going just from other conferences I've been at. So I'm not, but I know that's an area that people are starting to look into.
Great. And then Sarah Ryan says she's going to send you samples from Clear Lake, which is great.
Oh, that'd be fantastic. Yeah, that was one that I thought would be nice to get some microcystis from. So thank you, Sarah.
And then anyone else, please feel free to unmute and ask your question. Let's see. Shannon, you put your hand up.
Hi, Chris. Great presentation. I think, echoing what Hans was saying, that this work is really showing that it's a much more complex character, microcystis, than and that it's not a monolith either. And I think that we're seeing this, at least on the mitigation attempt side. Why water body managers want a simple solution, and they want kind of a one size fits all. And if they just put something into their management program, they won't have this problem, and board of directors will be off their backs and whatnot. But each one of these situations, the water body has very different characteristics. I can imagine that these would be quite variable even in the same water body, depending on if it's a drought year or a high precipitation year. Also the water levels, right? If they're shallow versus deep, and why in certain areas of maybe a lake that has several fingers or is more irregularly shaped than kind of what we think of as like a big oval, that would be certainly having its own niches even within a singular water body. So it's really important to tease out this basic science work so that we have a better understanding of what the implications might be for why blooms persist in certain areas, why they respond or don't respond to mitigation attempts, and how should we be looking from a risk management and water body water management side, because I think there's a inclination, particularly with public pressures, for water body managers, agency people, even state scientists, to want to either identify key sources, like it's always attributed to the sewage treatment plants or water treatment, or that it's this or that, and I really think it's multifactorial, and even within that, it's really what kind of populations you have. And what would be interesting too is to see, and I don't know how you would replicate this in a laboratory situation, but I'm wondering if in a lot of these cases it seemed to be repeat offenders, but also maybe behaving badly more one year than another, is that they're different strains. And does one major bloom set up the situation for another to thrive? And I've seen it in some of the lakes that have somewhat chronic or repeat HABs. One year we might have a huge microcystis bloom. Another year we might have less microcystis and more that would be akin to producing cylindrospermopsin or anatoxin A. And it just would be interesting to see how these varying growth optima conditions for microcystis kind of set the stage or may interfere with or exacerbate the growth of some of these other cyanotoxins that we see. So anyway, just thoughts to put in the back of your mind. As state people, we're always looking at the practical management application side of things, because the science is always really interesting, but we want to know where it's going to be applied to, right? So
yeah, absolutely.
Luck on your program. Well done.
Thanks so much.
Hans, looks like you got another question. Go ahead.
Yeah, just a quick question to follow up on the previous one. Chris, did you do any microcystin analysis on with your growth curves?
So you like microcystin production?
Yes.
Yeah, we haven't done that yet. We're so the experiments that Max did, this initial work, they were in 48 well plates, so we didn't have enough mass to probably do those, but we're trying to repeat them here at OU with kind of bulk conditions, where it'd be nice to get some of the microcystin information in those experiments where we'll have the material to do it.
Yeah. Also, if you still have DNA, from those experiments, you might be able to look at the gene copies too for microcystin production.
Yeah.
That would be cool because, one of the hypotheses about microcystin, why, when do they produce and when do they don't produce, is it may be related to growth rates. Because one of the hypotheses is that microcystin is actually produced to protect the cells from high oxygen levels when they're growing real actively.
Yeah.
As an ROS, harvesting molecule. So that would be really great. If you have DNA left, that would be something you could look at.
Yeah, absolutely. I mean, I think also doing a RNA, just qPCR, looking at microcystin transcription levels would be also advantageous there. So something, yeah, I think definitely think about, talk to Dave.
Yeah.
We'll see. Because
Dave,
I agree. Go ahead.
Yeah, I think Dave is interested in this question. We've talked about it for a long time, but it seems like if you have any DNA material or DNA material around still, it might be interesting to look at it retrospectively.
Yeah, absolutely.
Cool.
Thanks, Hans.
Other questions for Chris? All right. Okay. Well, thank you so much, Chris. Actually, I was emailing with a couple of people while you were talking because it's very interesting, and I'm gonna steal this person's question because I think it's really interesting. But you're pointing to that there's kind of essentially ecotypes, which I think makes a lot of sense. Probably the next question from a application standpoint is there some sort of genetic marker or something that could be developed or that you're thinking about where, in a management or monitoring application, you could look for specific problematic microcystis species or ecotypes?
Yeah, absolutely. So that's one of the details. I left a lot of our genomic work out, but we actually have proposed 4 pairs of marker genes that we now have primer sequences for that I'm beginning pilot work with, where we're hoping to develop that specific thing where we can just target these marker genes that are specific to microcystis that we know should resolve the same taxonomy, give us the classification, and would do just that, where we can sort of at a management level then identify what microcystis at a group level are in the lake and whether or not it's one of the more problematic ones or one that's just, kind of problematic.
Very cool. Yeah. Because high biomass is a problem on its own, with or without toxins.
Right. Just whether you're laying on what level of badness.
Yes, exactly. All right. Well, if there's no other questions, we're super grateful for your time and sharing your work with us. And I'll plug again that Chris is looking for microcystis cultures. And I know that many people in California have microcystis issues, and we should all send him bloom samples this summer for him to continue to do this work and have more California representation within this genomic analyses. So we'll go ahead and make sure that information gets shared around the community. Sarah Ryan's already taking you up on that. Yeah, so thank you so much, Chris. So I think the next item on the agenda is we're going to take a short break. We are ending a couple minutes early, but because I know people sign in and out of this meeting, we'll go ahead and reconvene at 10:30 still, so that anyone that's signing on for subcommittee updates and whatnot will be able to not miss that. So thank you again, Chris. Thank you, Amy, for your presentations. Very, very fascinating work from both of you. All right, everybody. Hope you had a good break, had some coffee, got refreshed. So we're gonna wrap up the last remaining items on our agenda. So we'll start with our apologies. I have too many windows open. with some updates from CCHAB subcommittees. And so let me see. First we have Carly giving an update on the benthic subcommittee progress. Carly, are you on?
Hey, yeah, I'm here. And I think Mike and Rich might be around, so if they want to jump in, definitely do so. But the Region 1 has started their study design. The benthic subcommittee met this spring. I unfortunately missed that meeting, but the idea was to kind of share with everyone the plan for what they're piloting this summer in terms of advisory signage. So they have deployed the spats. They've been doing the monitoring all summer. Thankfully, the levels have stayed pretty low, but we'll be reconvening the subcommittee again this fall to go over the results, share some insights, and then hopefully have the group come together on a more regular basis to start considering whether the current advisory framework works or if we should maybe consider amending parts of it. So if you are interested in participating, it's not too late. I'll throw my email in the chat and you can definitely reach out to me and we'll make sure that you get it as part of the group. And that's all I got.
Perfect. Yeah, a real quick update. Yep. So we, as Carly said, we're going to reconvene in the fall. So we'd be happy to have you if you are still interested. So next we have a mitigation subcommittee update from Hugh. So and then followed by Shannon. So Hugh, if you want to go ahead and start with your update.
Yes, hello, good morning, everyone. We've been meeting for a few months this year. We had a really good talk last month about a new biological beneficial bacteria treatment that some refer to as bug in a jug, but a little background on that. Similar product as the dry pellet kind of beneficial bacteria that you might have heard about. I know Aquafix creates one and people buy it online, but this is actually a treatment device that has a innocuous competitive bacteria to proliferate and compete for nutrients and try to outcompete cyanobacteria. So kind of have the possibility of starting a pilot next year. We have some background work to do before that, but yeah, so very interesting. It's been used for years in wastewater treatment. So the group is called N-Bioorganics, and they have a consortium of bacteria they use for industrial wastewater. They have a different grouping for municipal wastewater. And lastly, for freshwater, they have a different kind of bacterial grouping of beneficials. And I know Dave Karen is on the call, and Jamie was also at our meeting this past month. So if you've got anything to add there. Lastly, for updates, we have some newly installed oxygenation systems this year that we're hoping to get some updates on before the end of this year. How that's going, lessons learned, pros and cons, all that good stuff. So it's if Dave or Jamie have anything to add, that'd be great.
I think you covered it, Hugh.
Yep, great job. Thank you, Hugh.
Thanks, that's all I've got.
Awesome, thank you. All right, so Shannon, we discussed you're gonna give some updates from the illness work group. So I gave you screen sharing privileges if you would like to, you said you had a PowerPoint, so.
Yes. Okay, can you see my screen here?
We sure can, it looks great.
Okay, very good. Okay, so Karen covered a lot of this, so we'll just kind of breeze through here. So like I said, we're part of the California Interagency HAB-related illness work group, for those of you who are not as familiar. It's comprised of 4 key agencies. Our CDPH colleagues tracking California, they predominantly work up the human cases, do those investigations, although OWIHA also serves in that capacity. Domestic animals, that's generally OWIHA. Most of those end up being dog cases. Fish and Wildlife, CDFW, Karen, and then response and agency coordination, including water sampling, is generally under the Water Board. We meet at least every 2 weeks, but we're in constant communication via email. And that's how we make our determinations on which cases are meet the burden of evidence under the CDC's OHAB's definition for being HAB related. And if so, then they get reported into the OHAB's database. These are the key categories of collecting our information. Every illness report is accompanied by an environmental form. And so there's also requirements for that. These are just some details. If you're more interested, I can always send you more details. The illness determination is always made by the illness work group as a committee. So we're not a silo in any way. We all bring a multitude of skill sets. We have biologists, we have toxicologists, we have biostatisticians, we have research scientists or environmental scientists, depending on what phase of the committee we're in. So it's really a nice mix of those who are more the desk jockeys like myself, and then our field specialists. So it's really a nice group. And again, everything will fall into either being HAB related or not HAB related. Not HAB related ends up being the most frequent determination. And again, that's not necessarily because we've been able to rule it out, although with our expanded skill set, such as with the addition of CDFW's contract and Karen, we were able to make a definitive diagnosis in that case, which was Coy herpes virus disease. But in most cases, it's because we don't have enough information or we can't get a hold of the reporting party. And they might have put in an initial report that was missing some key information. And despite our best efforts, we've not been able to get more information. So we end up closing that case out. Obviously, with anything of the investigatory nature, there's some constraints and challenges. But one of those that we're really feel like we're making some ground on is that getting over this assumption that we have a HAB season. Really, HABs occur all year long, especially in California. And because of our wonderful weather, although when it's over 110, I'm not sure that's wonderful, we have people recreating and coming in contact with water. A few of our highlights from this year. So illness notification and reporting. This has really paid off our relationship with Poison Control Systems. So that's a direct contract with the WIHA. And we've worked with them to update some of their coding such that cases that get called in that may fall under a potential harmful algal bloom related illness, whether it be dermal contact, incidental ingestion, like they've swallowed either seawater or fresh water, or even consumed contaminated seafood potentially due to a HAB. Those cases get filtered and then sent up to a WIHA and CDPH, and with the reporting party, the caller's permission, and we'll work those cases up. A few recently, one, there was a harmful algal bloom incident of a Californian who had recreated in Zion National Park. And so we ended up calling that RP, getting more details. We were on a call earlier in that same week with the CDC's Community of Practice, which represents states who report to OHABs. So all HAB-related public health workers, essentially. And so we were able to do a one-to-one transfer and hand that over to the good folks at Utah Poison Control, and then also tell the Californian reporting party who they were going to be contacted by with an actual name. So we did a nice handover there. Other cases, it might be that they see warning signs for poor water quality due to a beach closure or a beach warning sign in Southern California due to high bacterial counts, not cyanobacterial counts, nothing HAB related, but it might be due to high E. coli or fecal counts of some sort. And so we have an opportunity to close that loop and do a little bit of outreach for other concerns. We also provide updates formally via email to our FHAB coordinators, representatives from all 9 water board regions, just to give them an update on what's happening and what our determinations are. So this is a nice summary. It's on our website from 2022. This is just showing that we have freshwater HAB cases representative of these counties. It's color coded by whether there were human and animal cases, not necessarily the same case, but reports at least that we determined were sufficient to meet our definition. So it ended up being 20 human cases, 3 domestic animals, dogs, and then 7 fish or wildlife incidents. Most of these are fish kills, but sometimes we have an odd turtle or bird kill or something to that effect. Just again, to dissuade the myth that we can have HABs any time of year, there's not a season. One of the things to keep in mind is that during the warmer months, it's more often that people are going to come in contact with a HAB because they're recreating in the water, they're boating, jet skiing, swimming, etc. So we're out hiking and part of that hike ends up being a water crossing, or their dog goes in or something like that. So it doesn't mean that we don't have HABs at other times of the year, but increase in water contact as a function of weather is a major component. We also have a new aspect to our website. So we have 5 good years of tracking, 18, 19, 20, 21, and 22. And so it has a pretty good coverage geographically across the entire state. And then right here, there's an accompanying text right above this map when you go to our website. And if you click on this active link here, that'll bring up a table so you can track by county how many human cases and animal cases were confirmed as meeting the definition for being a HAB-related illness. So that was at request at some of our water body managers throughout California. So that's helpful. So again, if you go to our website here, and you can easily just type in my water quality or HAB illness tracking, any of those kind of word combinations in Google, and you'll land on our site. Summary of marine HAB-related illnesses. We ended up having 116 last year. 113 were California sea lions. So again, the marine mammal impact. 3 were birds. 2 marine invertebrates and fish situations, and 2 human illnesses following water contact. Red tides of as so-called not associated with those that are generally seafood illnesses, but ones that are more of the water contact, like Lyngbia toxin. Those can be result in sort of an allergic type response following water contact when we have those bioluminescent red tide events. So that can be respiratory symptoms, and if there's incidental water ingestion, you can have GI symptoms too. So we had a couple of those last year. Just to bring you up to speed, we have brand new updated fact sheets. We do these every year as a review. And obviously we encourage these to be spread far and wide. Here we have our freshwater and then also marine. So please, these are the fact sheets, but again, they're pretty easy to find us. For the freshwater information, that's hosted by Water Board under My Water Quality. And then for the marine side, we host that on the OWIHA website. So that's just to help that information finding there. So where are we to date for this year? So here we sit in August, month 8. And on the freshwater side, we're actually doing a little bit better, knock on wood, than we have in previous years. We're still not out of the summer. We're still going to have triple digits again this weekend, at least in Northern California. So right now we have about 7 freshwater cases. I'm sorry, 7 human cases. 6 are active. 1 case is under investigation. These ones have been filed. They're predominantly in regions 5, 6, and 8. Again, we have 9 water board regions, but they are not unique as far as their geographic distribution. So some of these are huge, like Water Board Region 5. This includes also Clear Lake. So everyone out there working in Region 5 is really doing yeoman's work. So just keep that in mind. There was also the separate human case that I transferred to Utah. So just mention that there. There is a dog illness case under investigation, and then a few fish and wildlife incidents. On the marine side, I'm sure most of you know, and this is one particular area that I anticipate might have a question or two. There has been a major domoic acid bloom off the coast of the Southern California Bight, and that really started up in late May. So the primary suspect is Pseudonychia. That's a diatom phytoplankton that's always in the California coastal waters. Unfortunately, there was a perfect storm of events this year that included major upwelling. So that's when the deep nutrient waters from deep in the Pacific Ocean come up, mixing with some, increased temperatures, and then the ocean currents. So those 3 components coming together to really facilitate a major reproductive acceleration of the diatom Pseudonychia. Pseudonychia has many strains, like our friends Microcystis. Some are more prone or capable of producing toxins than others. The main toxin it produces, particularly the Seriata and Delicatissima strains of Pseudonychia, is domoic acid. Domoic acid is a neurotoxin. It doesn't necessarily have a bad impact on finfish, but it certainly does on mammals, particularly marine mammals. And I'm sure that most of you have seen the news coverage, particularly that over June and into early July of these mass stranding events. To date, we don't have an exact count of strandings. We have several rescue efforts that are being coordinated across the state, particularly our Southern California partners, SeaWorld, the Marine Mammal Centers in the Channel Islands, and also those in Southern California, LA, and Orange counties. They're really doing a major rescue effort. Number one, that's the first tier, so they can get these animals in hospital for domoic acid poisoning, give them supportive care. Hopefully they can recover and be released. Many of them, unfortunately, are too ill and don't recover and end up needing to be euthanized. The second tier is, of course, removing the dead animals from the beaches. The biggest concerns with respect to humans, regards to domoic acid, is the consumption of contaminated seafood. So we've also been monitoring, in collaboration with our partners at CDPH and CDFW, making sure that there are no significant elevated levels that exceed action limits for domoic acid. Right now, there's currently an advisory in Santa Barbara County to not eat any of the bivalve shellfish because of elevated levels of domoic acid. People that come in contact with waters that have domoic acids, pseudonychia bloom, it's generally not associated with an adverse effect. Again, it comes from eating contaminated seafood. So this is a bloom event where it wouldn't be characterized as what you might consider a red tide bloom. There's no color change, and there's no adverse effects really that we see as far as coming in water contact or incidental ingestion of ocean water that has pseudonychia or respiratory concerns. But it's really an unfortunate situation. One thing that's unique this time around is that not only the concentration of the bloom, but that it is so far offshore. And so because it's so far offshore, where the big bait balls of the anchovies and sardines are located, the primary fish species that these pinnipeds, the sea lions, and different types of fur seals and such, and dolphins are consuming. So the way that those animals are getting sick is because they're eating these large schools of contaminated fish, and then they're having brain toxicity, largely at the hippocampus. A crude way to think about it is that these neurotoxins punch holes in the hippocampus, and so they lose their ability to orient themselves for navigation purposes, and so they end up getting seizures, are very disorganized in their patterns, so they don't go back to areas where they can eat. They may strand on strange beaches. They don't get back to the rookeries, the little islands offshore where they have their babies, so the babies are sometimes adversely impacted because of starvation, because the mothers don't return. Also, too, if the mothers do return, domoic acid can concentrate in the breast milk as well as in the amniotic fluid. So the babies are being exposed when they're breastfeeding, but then also when if they are currently pregnant as well. And that's very often the case during this time of year, where sea lions might be pregnant and lactating at the same time. So they have a baby on board and a baby at their side. And so this has really been a catastrophic event. NOAA has led the charge as far as media and also organization of the rescue efforts. So we're giving them a wide berth, but once these final stranding counts, both for those that end up in recovery and release, those that are euthanized in care, and those that were just reported strandings and unfortunately deaths and had to be removed, those final numbers will be reported to the illness work group, and we will file cases appropriately with the CDC to OHABs. So it's just really unfortunate, but that's where we're going to be spending a lot of our time later this year. And then always feel free to collaborate with us if there's any way that we can provide you with literature or answer questions, or if you're interested in our process. And then in the various places where you have an opportunity to do outreach, just keeping everybody aware of best water practices, what to look for in a bloom, and of course, how to report a bloom or illness so we can follow up with them. And I'll take any questions.
Awesome. Thank you, Shannon. Do we have any questions for Shannon on her update? Not seeing any hands. I actually did have a question, if that's all right. For the 2022 summary, you'd mentioned that there were like maybe 100 sea lions. Is that 100 like beaches that had strandings, or is it 100 individuals? Because I
Individuals.
Okay, because there was kind of a significant fall time bloom event where there was more animals involved in that. So are you limiting that to animals that had like how are you confirming it as a DA case versus like?
So most of these numbers are coming through SCOOS and their
Okay.
counts there, although we did have a couple in the northern counties. And so when those have illness reports come out, usually it's 1 month in arrears. So we might get the June report in July. That's usually typical. When that report comes out, those are the confirmed cases. So in some of these stranding events, they may not be DA related. In most cases they are, but when they're not, it could be because they had a protozoal infection or something else that was confirmed through another diagnostic route, that it ended up being a stranding and an illness, but it wasn't HAB related.
Makes sense. Okay. Yeah. Appreciate that clarification. Any other questions for Shannon from the group?
I have one.
Go ahead.
Hello?
Yes, go ahead.
How are you disposing? How are you disposing of the dead animals?
So that is done by our marine mammal management partners. So I would defer that question to them. It's something I could make an inquiry about, but we don't actually deal with the actual animal cases, like the physical animals. But, you know, since this is something that they encounter quite often, just as a marine mammal medical facility, that would be a question for them.
Thank you.
I will say that one of the major outreach components of this current crisis, although it seems to be mitigating at this point, hopefully, is that people are obviously very emotionally struck by the immensity of this. And then anytime someone encounters an animal who is in distress on the beach, that's a very alarming situation. They oftentimes want to have an impulse to help. But really, these animals are already undergoing neurotoxicity, stress, they're disoriented, and Fish and Wildlife, as well as county officials, have been very strong in their messaging to give these animals their space. Unfortunately, it's the crowded beach season too, so lifeguards have been also brought into the effort to either kind of cordon off, if you will, big areas where animals may be stranding, to give them space so that either marine mammal rescue people can get in, or at least animals can have a somewhat calmer environment. Even some tents have been set up over animals that were in distress. Unfortunately, some of the larger males can't be taken into care because they're just too difficult to handle from a safety perspective, as well as just a size perspective. And so, you know, they're just kind of you know, allowed to have a peaceful area as much as can be done. So the concerns of late, particularly at the height of this event in June, were that people were having adverse wildlife encounters, where there were some reports of sea lions biting humans. And that's because, again, they're stressed out, people get too close, and then they're also having aberrant behavior because of the neurotoxin, where they might be more aggressive, or wandering around in areas that would be, you know, atypical for these kind of animals, and people don't know how to judge that behavior. So again, the stress was always to give them their space, as you would with any wildlife properly, unless you're a trained interventionist. Yeah.
Perfect. Yeah. Unfortunately, there were, I think, between 10 and 30 bites reported across the state. Yeah, very big, dangerous animals that people don't think are dangerous.
And again, you don't get DA poisoning, domoic acid poisoning, from a bite, but you can get all sorts of other bacterial infections, not to mention just the trauma of a bite. So again, people are cautioned to stay away. They're still animals.
Thank you so much for this update. Any last call for questions? And we can transition. So we're going to move to our final agenda item, which here, I'll go ahead and throw my camera on here. Final agenda item is our updates from our regional HAB coordinators. So let's go ahead and Carly, I don't want to pick on you, but if Carly Nelson is still on.
Yeah, I'm here. Pick on me, it's fine.
Perfect. Do you want to just start region by region? Because I'm not sure who's on to give their own updates or who's having you give them in there.
Yeah, I'm not totally certain either. So we'll just go in order. I think Mike is here. So Mike, I'll let you start us off for Region 1, which is our North Coast Regional Board.
Yeah, good morning, everybody. I'm going to share my screen, which is disabled. Jamie, I don't know if you need to allow me.
I do. Let's see, where are you? You've moved all over. There you are. Perfect. There you go. You should have access now.
Awesome. Thank you. Okay. Can you guys see that okay?
Looks good.
Yeah, so my name is Mike Thomas. I'm the FHAB coordinator for the North Coast region. I'm just going to give you a quick update of what's been going on so far this year. So we have some routine monitoring going on. This is being conducted by Big Lagoon Rancheria, as well as the tribes in the Klamath Basin. This includes biweekly monitoring. To the right there, you can just see a picture of the Klamath Basin and the various sites that are being monitored. We also have some monitoring going on in the Russian River. Sonoma Water is conducting some pretty extensive surveys, and Sonoma County is also doing some visual assessments. We also are doing the pre-holiday assessments. Listed here are the various water bodies and the sampling entities that are conducting those assessments. Most of the water bodies are being sampled from 4th of July, Labor Day, and the extension or post-holiday period. Some other efforts going on in the region, partner monitoring. This is occurring at Big Lagoon. This is in addition to the routine monitoring that they are doing. We're just collecting some extra data on additional cyanotoxins and cyanobacteria. And this has been occurring since April and will be going through October. Also, Carly had mentioned the benthic monitoring that we're doing in the region as part of the benthic subcommittee efforts. We are evaluating and implementing our tiered monitoring approach, which involves SPAT deployments, visual assessments of cyanobacteria percent cover, as well as benthic mat collections. This is occurring in the Eel, South Fork Eel, Russian, and Navarro rivers. Some other studies and research. We are working with University of Nevada at Reno and CSU San Marcos, looking at the spatial extent of benthic cyanobacteria and cyanotoxins. This is occurring in the salmon and South Fork Eel rivers. We are also participating in the US EPA ROAR project, where we are developing standardized benthic sampling methods. And we have 2 sites participating in this nationwide study. Some current postings in the region, not as many as I think as last year at this point in time, but those are listed here. Most recently, it was the warning recommended for Iron Gate Reservoir. And as always, just want to thank our partner agencies listed here. And many of these we have been meeting one-on-one this season to discuss developing their own FHAB programs and any monitoring approaches they may want to take. So that, I'll stop sharing. And yeah, pass it on to the next.
Yeah, sounds good. Thank you, Mike. So I think the next, we'll just go numerically. So that'll be region 2.
And that I think might be me again. Region 2 has been very busy. As some of you may know, there's currently a bloom in the San Francisco Bay. The heterosigma is back again this year. Hasn't made as much waves yet, which is great. But so they've been doing a lot of incident response with that. And as well, I guess something to highlight while I have you all is we are now putting some of these blooms, the marine blooms that we are getting reported to us on our HAB web map. So if you are getting reports, you can send them our way for any observations. And then as well, I think Karen might still be with us, but if you receive any reports of any fish deaths, then you will send them to Karen Atkins and the iNaturalist. Yep, she's going to be triaging all of those. But as for some other updates with Region 2, they're also participating in the holiday assessment, and they're monitoring 5 sites for Memorial, 4th of July, and Labor Day. Berkeley Aquatic Park, sorry, I'm trying to remind myself. Marina Lagoon, Shoreline Lake, Napa River, and San Geronimo. And for the last assessment for the 4th of July, they did get a caution for Marina Lagoon, but for the rest of the sites, it's been pretty quiet. And with that, they've been busy kind of just doing some incident response. But as I said, most of their efforts have been going to this new bloom that's been developing since we found out about it at the latter part of last week. Oh, and I will put some information in the chat with that. They did put FAQs up on the webpage for more information, as well as on our HAB web portal. There is information now there as well to direct the public as to where to report some of these observations, and then the iNaturalist link as well. So keep pointing folks to us. Thanks.
Great. Then do we have anybody from East Bay on the line? I thought I saw somebody earlier if they want to give an update. Perfect.
Hi, yeah, Sabrina Sexton from East Bay Parks. So it's business as usual for us with our weekly monitoring this time of year. We did actually just, in terms of freshwater HABs, we just downgraded our advisory at Horseshoe Lakes at Quarry Lakes. So that is now a caution advisory. It had been in a danger advisory for a couple months now. We did just complete posting red tide caution advisories as well along all of our shoreline parks. And yesterday we just had another active treatment at Lake Temescal to help abate the current bloom there. And that's about it. Thank you.
Thank you. And then anyone else from the Bay Area that wants to give an update? I don't think so. I see anybody. Let's see. So then moving to Region 3, Carly, I think that's you again.
Yeah, I know. They're all busy with the field season. So I'll be, that's the Central Coast Regional Board, and that's Melissa Daughtery. And they participate really heavily in our holiday assessments as well. So they're out sampling 6 different sites, sorry. She put them in the chat, but for those of you who aren't familiar, Oso Flaco Lake and the Creek, Lopez Lake, Laguna Lake, Lake Santa Maria, Margarita, Lake San Antonio, and Lake Nacimiento. And for those, I want to say Oso Flaco Creek and Oso Flaco Lake both triggered caution advisories last 4th of July with that assessment. And with the rest of them, she had recommended general awareness because there were some cyanos via microscopy, but it was below our toxin thresholds for caution. So it's been a relatively quiet season for us so far, which is great.
All right, so moving to Region 4, which is LA region. Looks like Emily's signing on.
Hi, yeah. No big updates. I mean, the usual harmful algal bloom activity at Pyramid Lake, which Brianne sends out updates in her monitoring emails from DWR. And I believe that Pyramid Lake has 1 location at danger advisory right now, so that's kind of high. And then we've received a couple notices from the public. Unfortunately, one of them I was emailing with Carly earlier this week is pretty far, a couple hours away from me in Santa Paula, north of Santa Paula, and people aren't including GPS coordinates in their submissions, but this person included pictures and it looked like a lot of filamentous algae. And then we had another on the Arroyo Seco River near NASA's Jet Propulsion Laboratory, which is a really popular recreation area. And I did a site visit and it was a lot of filamentous algae there. So far, that's all we've got this season.
Thank you, Emily. Anyone else from the LA area that would want to provide an update? All right, so then moving numerically would be Region 5, Central Valley Regional Water Board.
Hi, I thought I'd introduce myself. My name is Dana Schultz, and I started as the new Central Valley Region 5 Freshwater HAB Coordinator just this past June. So still getting my bearings, but diving right into the field season. Prior to joining the water boards, I worked for 5 years at SQUIRP, most recently as the research coordinator for the biogeochemistry group, which includes Jamie Smith's HAB program. And before that, I got a master's studying marine HABs at UC Santa Cruz. So for the Central Valley, I thought I could give you an update on some of our partner monitoring efforts, advisories we have on the water board online map, and some of our holiday assessment efforts. So for partner monitoring, we have a program going with Restore the Delta. This is a continuation of work that Karen Atkins and Janice Cook on the water board side of the partnership got started last year. So this summer, Restore the Delta and Spencer Fern and his staff and interns are going out to 7 sites along the Stockton waterfront and San Joaquin River. They're going out weekly to collect water quality measurements, and they're also doing visual observations for blooms as well as microcystin test strips. And then monthly, they're collecting samples to send to Ben Genetics for microcystin nodularis ELISA analysis and analysis on a suite of toxin genes. And I can report out on that the most recent sampling that went to Ben was the week of July 10th, and so far we have not had microcystin detects in that area. Another partner monitoring program in Region 5 is in partnership with the state parks along the North Fork of the American River. So State FHAB, Marissa and Carly are leading this effort. And the state parks are collecting samples to analyze, sent to Ben to be analyzed for microscopy, as well as a suite of algal toxins. Their most recent sampling was July 18th. They went to 4 sites. There were algal mats present at 3 out of the 4 sites. And each of those algal mats had potentially toxic cyanobacteria found via microscopy. Only 1 sample had detectable anatoxin A, and no other toxins were found in any of the other samples. Moving on to advisories we have on the map in the Central Valley region. We do have algal mat alerts for those 3 samples from the state parks where there was potentially toxic cyanobacteria found. Our danger, warning, and caution advisories mainly exist at Clear Lake. And we do have a couple visual observations on the map at Los Vaqueros Reservoir, Delta Coves, Brookside Lake, McKinley Pond, and Millerton Lake. For the holiday assessment work, we're about to gear up for the Labor Day holiday assessment. We have funding to support a handful of samples at Clear Lake, in addition to the regular monitoring that they conduct. Swamp staff from our Fresno office, that would be Akira, are going to go out to Hensley and Eastman Lakes. And then we also had funding to support holiday assessment at Kuya Lake and Kuya River. They haven't been able to sample so far because of water levels and flows, but hopefully we'll see if we can get something done for pre-Labor Day. And then the last thing is that Region 5 will be joining the EPA ROAR effort you've been hearing about, sampling benthic cyanobacteria at the site in the American River. That's it for me.
Great, thank you, Dana, so much. So anyone else from Region 5? I think Sarah Ryan typically gives an update for Clear Lake specifically, but she unfortunately had to sign off for a different obligation. So if you have questions about some of their observations, go ahead and get into contact with Sarah. Beyond that, if you're in Region 5 and like to give an update, go ahead and speak up. Otherwise, we can move on to the Hutton region with Sabrina. Awesome. Thanks, Sabrina. Go ahead and.
Okay. Good morning, everyone. I'm Sabrina Rice. I'm the coordinator for the Lahontan Water Board office. And yeah, I'll just give a brief verbal update. We have also had a bit of a slower start to the season, luckily. But we have also been working on the pre-holiday assessments. We monitor roughly 20 water bodies during that time frame. And most of them this season we're doing Independence Day, Labor Day, and a post-holiday, just because we tend to have a little bit of a later bloom season. And so yeah, we're continuing with that. We have a few new partners who are helping with that as well, so that's always great. And then in previous seasons, we did 2 special studies at Red Lake and the Tahoe Keys. And this year we are no longer really doing the special studies. We're doing still some monitoring in those lakes or those areas, but it's just a bit dialed back from previous years. So like the Tahoe Keys are no longer doing the nutrient monitoring, but the Tahoe Keys Property Owners Association does do a lot of monitoring in that area. And, you know, they're involved in a few other projects where they have to do monitoring for some of their permits. And then Red Lake, we're still monitoring in the lake itself, but not so much the tributaries. And then we are trying to start up a new partner monitoring program in Inyo County. That's on the Eastern Sierras, and this is with Inyo County Environmental Health Department. They are a great partner. They have participated since, I want to say 2016, in our just monitoring and in the pre-holiday assessments. And so they're trying to just do a little bit more routine monitoring in Diaz Lake and Mill Pond, which is in Bishop. And then they might be monitoring some other areas when they have access, just due to the larger water year this year, or snow year we had. They haven't had access to certain areas, but they're trying to get down to Pleasant Valley Reservoir and some areas along the Owens River. And then for advisories, we have had a warrant. I'm going to just try to go north to south. So in Alpine County, Indian Creek Reservoir, we monitor this water body with South Tahoe Public Utility District. They have just reached a warning level, and they monitor roughly monthly at this lake, partially during the pre-holiday assessments, and then also just on their own when they see visual indicators. The Tahoe Keys just reached a caution level, just again, part of their routine monitoring. Lake Barron, that is in a smaller kind of park near South Lake Tahoe, and that reached caution. It was slightly below the actual caution level tier, but just due to some visual indicators and the level of rec use seen at that lake, we decided to go with a caution. Talac Creek, that is a creek that flows into Lake Tahoe at Baldwin Beach, if anyone's familiar with that beach. This area, we are just keeping it up just for consistency. The Forest Service has decided to keep up signs just to be safe, but the monitoring showed that that was below any caution tier. Crowley Lake is also at a caution. That is in Mono County on the Eastern Sierras, kind of near Mammoth. And then lastly, we have Green Valley Lake in San Bernardino County. That one right now is currently off visual indicators, but we are planning to monitor this week to actually determine what the toxin levels are, if any. And that's it for me. Thank you all.
Thank you, Sabrina. Super appreciate that update. Anyone else in the Lahontan region that's doing monitoring online that wants to do an update? And if not, we'll go ahead and move to region 7, which I'm not sure if Carly, that might be you again.
No, I'm here.
Oh, you're here. All right. Thank you.
This is Jeff over at Region 7. Yeah, right now we're only conducting HABs monitoring for incident response and pre-holiday assessments. So we've really scaled back. But we did have a fish kill in July at one of our county lakes. So I really wanted to hear more from Karen, but I guess we didn't have time for QA. I did have the perfect chance to collect dying fish for a necropsy, but we didn't have anywhere to send them. So that was a missed opportunity. We also could have maybe saved some money on pesticide analysis, if we had a little bit of preparation and training. But we're just not equipped to respond to fish kills, but I think we should be. I'm teaching myself right now, and I know that Fish and Wildlife typically performs those investigations, but I definitely think the regions should establish some level of training and set up resources for responding to kills and maybe collecting better data when we respond to a kill. But anyway, that's pretty much the extent of our HABs work right now. Thanks.
Thank you so much, Jeff. And we're doing really good on time at the moment. I don't know, Karen, if you want to just respond to that or handle that offline.
Sure, yeah. It is my intent to hopefully get to train all the FHAD coordinators at some point. And I would love to work with you, Jeff, more. We are still kind of like I'm still getting my feet under me a little bit in terms of just getting even supplies to our staff. So in terms of getting fish sampling supplies, that's still coming down the pipe and might not happen this summer. But yeah, I'd love to work more with you and with the rest of the FHAB team at Waterboards too.
Great. Okay, so if there's no one else in region 7 that's doing partner monitoring or anything that wants to provide an update, we'll go ahead and move to region 8 in Riverside.
Hey, good morning. We've mostly been involved with pre-holiday assessments at Lake Hemet, Lake Elsinore, and Big Bear Lake. We've also been collecting samples for eDNA monitoring. And so far, I think we're only at caution levels of cyanotoxins. We have been working with the City of Lake Elsinore, along with Carly and Marissa, to encourage additional monitoring. So far, they've been monitoring semi-frequently at several sites around the lake, and they've been communicating that to the public. But they've kind of dropped off in communication recently, so I have to follow up with them. We've also been a little concerned about there's a inflatable splash park that has been installed in Lake Elsinore, and there's been some communication from the operator that would indicate that there's no harm in entering the lake, but Lake Elsinore has a consistent bloom throughout the year, and so we always recommend that the lake be at caution levels at the very least. So we've been trying to figure out what to do with that. I also need to follow up with the city to see what they're doing with the splash park, but I know it's operational. So yeah, I think that's our update.
Thank you, Barbara. So moving down, we'll go region 9, and I think Carrie, you should have screen sharing privileges. Looks good. I think you just need to unmute yourself.
Oops, sorry about that. So just a quick update for Region 9. I'll start with getting my slides to move ahead. There we go. So we have 4 reservoirs that are currently posted, and most of these are actually being monitored by other entities, which is great. So a lot of our reservoirs are operated by the city of San Diego, and then various water districts. So the first one on the top left is Diamond Valley Lake, and that's Metropolitan Water District. They currently have a caution level, and that's due to microcystins. And I just popped up their website so you can see that they have notification on their website as well as signs physically at the reservoir. And then top right, Lower Otay, that's operated by City of San Diego. They do all the testing, and it's currently at a caution as well for microcystins. Bottom left, Lake Henshaw is typically our problem reservoir, and it's pretty much posted year round, even though this year they've treated a few times, and the levels right now are actually below the caution level, but they just continuously keep the signs up. And then finally, the bottom right was sort of a new partnership for us, which was kind of neat. Lindo Lake, it's a county park, and there are volunteers from the NOAA Phytoplankton Monitoring Network that are out sampling there, that were the ones that had notified us that there was a bloom, and there are signs physically at the lake now. As far as our activities, we are doing the pre-holiday assessments. We have 6 streams that we chose that are high used for recreation a lot, and so far so good. We've had all non-detects in those, and we're ramped up to start for the Labor Day sampling soon. And then the other studies that we're doing are 2 kind of mini special studies, and one is a screening of our high quality streams that we've been out doing bioassessment for years now, and we've noticed that some of them do have a lot of cyanobacteria that we have noticed in the DNA sampling, and as well as physically while we're out. So we were just curious what's going on with that, if toxins are something that we need to worry about. And then the other study is a continuation of a high agricultural use watershed screening study that we did in 2021. So we're going back to the sites that had the most and the highest toxin hits in the spat, and pairing that with some other sampling too, which we just started that sampling. So here's just a quick little update of the high quality streams. So we see nostoc a lot, and we have other cyanobacteria, like I said, that have been identified in the DNA sampling. And so what we did with those is we paired spat deployments with our algal composite that are collected during routine bioassessment. And then we also did some targeted benthic mat collections. So far, what we found with those, with the benthic mat and the algal composites, we aren't seeing toxins. But in the spat, which was kind of interesting, they all had antitoxin detected, low levels, but they all had that, and then one had microcystin. So we'll be working through that and trying to make sense of what we're seeing with that study. And then our follow-up study at the high agricultural watershed streams, we chose 3 streams that had the highest concentrations and most frequent spat toxins from 2021. And we're deploying spat weekly, and we're also collecting nitrogen and phosphorus samples, and then targeted benthic mat samples if we get spat hits. So far we've only had the first set of spat out, and I'm going back tomorrow to collect those. So we'll see what happens with that. And that's all I have for Region 9.
Perfect. Thank you so much, Carrie. That's an update to see what you're working on. And thank you for everyone. Just want to make sure, are there any other monitoring updates that I missed? And anybody else want to, I don't want to put Randy on the spot, but Randy, you put a really
I was gonna jump in.
You were gonna do that. Okay.
Yeah, Randy, I didn't really prep you for this. I wanted to make 2 comments before I send it to Randy. One, just that the regional coordinators underplay and are very humble about the work they're doing this summer. And just to give you guys an idea, for the July assessment, most of them went out and sampled themselves, and they sampled over 80 sites. So it's really impressive, and it's a lot of work, especially when a lot of them have competing other sampling priorities. So they just deserve a lot of accolades for that. And it's awesome because then we can do a lot of press for that and get it out on Twitter and different things to like really make the public aware. And then secondarily, you might have heard this ROAR study mentioned by Regions 1 and Regions 5. So that's a collaboration we're doing with US EPA, with their Office of Research and Development, to kind of pilot out some different theories and different methods for benthic collection. And once again, Mike and Rich are underplaying their role as well. But US EPA kind of was seeing what we were doing in California and realized that we might have some good morsels of information and education to share about what we're seeing here. So it's a really good opportunity for us to pilot a few water bodies with some of these different methods and see what works and what doesn't work. And we will be, we'll circle back with the benthic subcommittee with that, but we haven't really done a lot with it. I just wanted to kind of give you guys a little bit of a knowledge base as to what that's about. And with that, I would love it if Randy would do a quick demo, or I can. Randy, it's up to you, of some updates to our satellite tool. It's brand stinking new in the last couple of weeks, and it's super timely with the San Francisco bloom. So we're really excited to have this tool up and running.
Yeah, happy to share my screen. It's currently disabled, so Jamie, I'm going to need you to help with that. Yeah, Lawrence Sim and I from SFEI have been working closely with Carly and Marissa to build some additional functionality into our harmful algal bloom remote sensing tool. So I presented on this recently at the latest comms webinar, but the general idea, if folks are not familiar with it, we have a tool that can assess cyanobacteria concentrations using remote sense data, and it's been up for quite a few years now, but we are now using the same satellite data from Sentinel-3 to also generate estimates for chlorophyll A. And so there's a lot of value here in being able to both determine what is a cyanobloom and what's just a bloom of other algae, but also just to look at general, primary production and biostimulatory conditions. So there's a lot of value in being able to have both of these data sets be complementing each other. So if we look at Central Bay right now, we're not seeing anything from a cyanobacteria perspective. But now with this new feature, we can go and we can look at chlorophyll as well. And we will see, again, kind of a heat map color pattern going on in Central Bay and maybe a little bit more so up in San Pablo Bay. If we compare back to the bloom that people are likely familiar with back in August of last year, it was definitely a different story from a chlorophyll perspective, much higher concentrations, which we may still get to at this point, but you can see the general difference there. And then but when we were looking at this tool last year, when it just had cyanobacteria, even during the peak of the bloom, pretty much nothing. So again, this is referring it to a non-cyanobacteria bloom with the dinoflagellate. So a lot of value there. We also have the ability to look beyond our kind of standard so far has been a 10-day composite, which pulls the highest concentration over the last 10 days for each pixel and puts it all together as a great way to remove influence from clouds or glare where we would have a gap in the data. And so, but we feel that's the most valuable tool for data set for most people. But we've added the ability to look at just a single day composite as well. So that lets folks kind of see a little bit more clearly of like the most current conditions. And especially for folks going out in the field who might need to know, where the bloom is today. So I think that's really a helpful way to look at things. So if we go just to a few days ago, what we'll see right here in the 1-day chlorophyll images looks like probably some clouds kind of pouring through the Golden Gate. We'll just advance here a little bit, and we start to see a little bit of higher concentrations on the edges of the bay, and a little bit more of a bloom picking up in San Pablo Bay, and then again, kind of more clouds yesterday, unfortunately, but we're seeing just some imagery that is really helpful in evaluating blooms in general, help confirm when it's cyanobacteria or not, and so it's really something that is, I think, very useful to pretty much everyone who's on this call. With the caveat of it works pretty much in the large lakes and water bodies only. So for folks working in smaller lakes, there's really a lot of limitations just based on the pixel size and our confidence in ability to look at just a few pixels in a small water body. It's a little challenging, but anyway, lots of value here. We are going to be looking at some other features to add now that we've got the chlorophyll data set included. So additional statistics to generate, possibly some, trend evaluations built into here. So, this is the type of thing that we would love to get input from, remote sensing experts or, folks working on blooms to look for what value you want to see. Are there certain stats you want to generate, trend analysis, that type of thing. So, looking forward to getting your input, either directly through us or through the Remote Sensing Technical Advisory Committee that Jamie Smith is helping run. So, looking forward to making this tool more valuable for you. I'm happy to take any questions that folks have.
Any questions for Randy?
And I put the link and my contact info in the chat if anyone wants to reach out later. That'd be great.
Great. Okay, so any other announcements or monitoring updates or questions while we're all on together? Or suggestions for topics for our next quarterly meeting? Okay, sounds, I'm hearing a resounding people want to go to lunch a little early, which is great. And I'll go ahead and, sorry, put my camera back on. Oh, so with that, then let's go ahead and we'll go ahead and wrap up today's meeting. We appreciate all of our speakers and updates from all of the FHAB team and partner monitoring groups. As Carly mentioned, I just want to echo that is so much work, and it's so great to hear what everyone is doing when we get together as a community. And welcome again, Karen. We're excited to have you as a co-chair. And I guess I mentioned, unfortunately, Sarah had to sign off. I do see real quick, looks like Shannon, if this is a new hand, I don't know if I just missed it from previously, go ahead and we'd love to hear from you.
Yeah, I just wanted to extend the real appreciation for the CCHAB co-chairs. I know that some of you are less seasoned than others, but I just think it's really a testament to this group and the wide range of expertise and interest. And I'm always impressed by the speakers that we get. They really are wide ranging, and everything from messaging and management to basic science. And I think it's really critical to represent that spectrum, because it really is exemplary of the multitude of layers and challenges that we're encountering as with our varying roles with respect to HABs in California in particular. So anyway, just really appreciate that effort. I know it's not a minor one.
Really appreciate those comments, Shannon. Thank you. And as Shannon mentioned, we're always happy for suggestions on speakers or projects. Go ahead and feel free to email any of the co-chairs with that. And so I realize I'm probably standing between you and lunch, so we'll go ahead and wrap up, and we'll look forward to seeing you in the fall in October for our fall meeting. Thanks, everybody.
Thanks, Jamie.
Thank you.
Thank you.